Okay, so decided to go through Unit 3 again with you. Wasn't comfortable with the idea that we didn't get to all the material that I wanted to in class, and I know that the immune system was a unit that people did struggle with in A and P, so taking for granted that you know all the components might not be the best idea. So I thought if I went through it again and posted this for you, that you could go back through the material kind of on your own pace, and then at least you will have seen all the material that I had intended to get through. So and I'm going to start from the beginning, but I'll go through fairly quickly the material that we've already covered, and I'll throw in a few points that I thought were the important points, but we'll get to at least to the point where we left off fairly quickly. Okay, so just make sure you understand the difference between specific and non-specific immunity. So the barriers versus if things were to enter the body and you have something that targets the invading substance or microorganism that's non-specific, doesn't really care what it is, versus your immune system that we call adaptive immune response. They target a very specific antigen in the body. So that first line of defense are, again, your mechanical barriers, so the tears, secretions, gastric juices, your skin. Second line of defense is when things do manage to enter the body, but again, it's not specifically looking for one antigen, so it includes phagocytosis, the inflammatory response, interferons, and the third line of defense, which we call our specific or adaptive immune response, which involves the T cells and the B cells. Now, the immune system components I talked about briefly. The skin and mucous membranes, just to add a couple things to that, there are antimicrobial peptides known as defensins that you might want to make sure you know. And there's also IgA antibodies, which are common in saliva. The mononuclear phagocyte system includes dendritic cells, monocytes, and macrophages. The lymphoid system, which the bone marrow is a part of, includes your primary and secondary lymphoid organs. So the bone marrow and thymus being the primary lymphoid organs, and the spleen, lymph nodes, Peyer's patches, tonsils are all part of the secondary lymphoid organs. Good idea for you to know what the different white blood cells actually do, at least in a general sense. Just as an example, neutrophils are most common and or most numerous and function in fighting off bacterial infections. And knowing which ones are part of the primary versus, the specific versus non-specific immune response as well. There are also the chemical mediators, which are not necessarily cells, but they are very important components of controlling and directing the immune response. So the complement proteins are the 20 plasma proteins that enhance inflammation and chemotaxis, mainly synthesized in the liver by macrophages and neutrophils. The kinins are the small polypeptides that cause powerful vasodilation, and I use bradykinin as a good example. The clotting factors function in blood coagulation, so they form the insoluble fibrin strands that form that mesh that blocks off the site of injury, so you stop bleeding. And cytokines and chemokines, which there's a large number of them, and they're in your textbook on page 171. But they're the polypeptide signaling molecules, so they affect the function of other cells by stimulating surface receptors, really. Mainly produced by T cells and macrophages, and some of the specific functions include chemotaxis, which are really the chemokines, so they attract other white blood cells to the site of injury. Antiviral factors, interferons, are an example of that. They function as mediators of inflammation. They stimulate the production of more blood cells, so hematopoietic factors, and they activate their activation signals for white blood cells. Now, inflammation is a non-specific body defense that responds to any type of tissue damage, infection, or irritation. Main four cardinal signs are the redness, the swelling, warmth, and the pain. And you've probably heard the term itis. Itis describes any type of inflammation, so whether it's your tendon or your stomach lining. Now, with the inflammatory response, one of the first things that happens, you want to get more blood to the area, so you get increased vascular permeability. So under the certain what we call vasoactive chemicals, the blood vessels, the capillaries become a lot more permeable, and this allows a lot of the immune cells to get into the site of injury and carry out their function. So you probably remember the terms margination and diapedesis. That's the way that white blood cells kind of adhere to the blood vessel wall and then squeeze between the epithelial lining to get into the interstitial space and target the infected area. This is where chemotaxis is important. And of course, when the white blood cells get to the area, the neutrophils and macrophages, they carry out their phagocytic function to try and remove the antigens, cell debris, and unwanted substances. So the diagram kind of shows those three steps. We talked a little bit about the capillary exchange, which has to do with the increased blood flow to an area. So normally with the capillary exchange, you get blood flowing to the capillary network, and then mainly things like water, electrolytes, specific nutrients will leak into the interstitial fluid and transfer materials. And the one thing that you don't get in a normal capillary exchange is large cells and proteins leaking out of the capillary. Now, in an inflammatory response, you do get proteins and cells leaking out into the surrounding tissue because of the increased permeability of the capillaries. And what happens is you also get a fluid shift when this happens. So you get a lot more water flowing out, because as proteins flow out, water kind of follows to try and balance things out. And that's why you get the swelling. And if you can imagine, if you have widespread or massive inflammation, you get a lot of fluid shifting out of your blood into the surrounding tissue, and that's where we start to get dangerous, where you start losing blood volume. Go through the manifestations of local versus systemic inflammation. So again, the redness, the heat, the pain, the loss of function, sometimes, not always, but loss of function. For example, if you have swelling in a joint and it doesn't move properly, well, then you've lost function. And specifically with swelling has to do with exudate, which is material, the fluid which enters the interstitial space, and it can vary. So if it can be watery, the serous fluid, it can have high fibrin contents, the fibrinous. Purulent is the pus fluid, so it's thick, it's that the yellow colored, it has a lot of high content of leukocytes, which is what gives it its coloration. And the abscess is a pocket around a solid mass, so you're probably familiar with a tooth abscess, and anything that contains bloody exudate is hemorrhagic. And systemic inflammation, some of the manifestations include increased internal temperature, so fever, neutrophilia, that feeling of fatigue, loss of appetite, muscle catabolism, so the wasting away of muscles. And if I just want to focus on a couple of these specific cytokines, the TNF alpha and the interleukin 1, they both act on the brain to increase body temperature, induce sleep, so that's where the fatigue comes in. They suppress appetite, which is where the anorexia comes in. And all three of these, TNF alpha, interleukin 1, interleukin 6, they all cause the liver to produce acute phase proteins. So the C-reactive protein and something known as serum amyloid protein. And what also ends up happening with these acute phase proteins is it increases the serum fibrinogen, which is a way of measuring the erythrocyte sedimentation rate. So the more fibrinogen you have, the quicker the red blood cells will sediment if you spin them in a centrifuge. And so if you have a higher ESR, then it indicates that there's inflammation somewhere. Which brings us to the changes in the blood. Of course, if you have inflammation, your white blood cell count should be over 10,500. That's kind of the threshold. It's for leukocytosis. The elevated CRP, the C-reactive protein, which is a good indication of inflammation. The ESR, or the erythrocyte sedimentation rate, will be elevated, which I just talked about. And you can even look for specific enzymes or isoenzymes in the blood, and these can indicate that there has been damage to tissue. So AST is a good example, which you'll find in someone with liver disease or someone who's had a heart attack, and same with creatine kinase. You'll find that in someone who has had a heart attack. So if you measure those in the blood and you find them, then it indicates that maybe the person has had a myocardial infarction. And this kind of summarizes what we've just sort of talked about with regards to inflammation. And again, you can take your time, and if you want to get a better look at this diagram, I don't think this is from your textbook specifically. I got this from somewhere else. There are complications that can arise in inflammation, so it is a normal response. But if you want to treat inflammation mainly because of the potential damaging effects of the inflammatory response. So you can get infection because of the leaky blood vessels, increased permeability, increased likelihood that something's going to breach the system and cause an infection, and even spread an infection from one location to another. After a long period of time, you can get ulcers, which can, if they're not properly managed, can become necrotic. Irritated muscle tissue can spasm and cause pain, and of course we talked about the local complications. So depending on where the inflammation is, like a joint, for example, you might lose function in that area. This is an example of diabetic foot, I believe, an ulceration that turned necrotic. When it comes to treating inflammation, many of the treatments aim to do several things, and those include reducing pain, reducing fever, so antipyretics, and counteracting the actual inflammatory response. And whether it's aspirin, Tylenol, they carry out all those similar functions. The non-steroidal anti-inflammatories mainly counteract the inflammatory response, and so do the glucocorticoids or corticosteroids. A couple of examples of corticosteroids: prednisone, hydrocortisone. They're all related to cortisol. And what they basically do is that they suppress the cells responsible for inflammation. Alternatives, if you don't want to use drugs, the resting, icing, compression, elevation, all these strategies aim to reduce the damaging effects of inflammation and allow tissue to heal at a moderate or a controlled rate and regain function. We talked about the types of healing, so it's possible that that tissue the damage resolves and the tissue starts to, the cells recover and function normally. So that's when the damage is fairly minimal. It's possible for cells to regenerate depending on the cells and their location. Some regenerate very well, like the liver. Others, like the central nervous system and heart cells, do not regenerate well at all. Or in cases where you don't get good regeneration, like the heart and the brain, then you get replacements. So the what was functional tissue now becomes non-functioning scar tissue, and that can obviously have permanent, lasting effects. Talked about the difference between healing by first and second intention, and the main difference being that in first intention, the wound is clean and the edges are fairly close together, and you end up with a much smaller scar. And the second intention is, when there's a much larger break, the inflammatory response is maybe a bit more prolonged, and you end up with more scar tissue and a larger scar forming in the end. This diagram kind of goes through the two types of healing, first and second intention. Again, if you want to take the time and just sort of look at them in a bit more detail, then you can do that. I wanted you to make a list of things that promote healing and things that delay healing, and I think we came up with a number of things. So those that promote healing include good nutrition, so vitamin A, having vitamin A and C in your diet, and good proteins, adequate hemoglobin levels, having good effective circulation, the wound itself being clean and undisturbed, no infection or further trauma to the site. I mean, those all will promote healing, whereas those that delay healing, the older you are, the worse your nutrition, the worse your hydration. If you happen to be anemic, so if you have low hemoglobin, if you have circulatory problems, if you have other disorders like diabetes or cancer, if you irritate the area that's trying to heal, things like prolonged glucocorticoid use, if you're going through chemotherapy, all those might delay the healing process. And specifically with regards to scar formation, you can get a number of complications. So you can lose function, especially if the scar is larger than one is larger than the other. You probably lose some function in that area. You may have noticed where if you do have a scar on your body somewhere that you don't have the same sensory function in that area. Contractures where the scar tissue is not elastic and tends to kind of shrink over time, and it can reduce your range of motion. Adhesions include bands of scar tissue that join two surfaces together, and they're common when you have bowel adhesions. Hypertrophic scar tissue is where you have a large overgrowth of fibrous tissue, and you end up with something known as a keloid. And of course, because of the impaired blood flow, you can end up with an ulceration, which can end up leading to necrotic tissue if blood supply is not returned. Antigens, also called immunogens. I don't think I need to spend too much time to talk about this, but this is what is on the surface of cells and what allows our body to kind of recognize our own, whether it is a self-antigen versus a foreign antigen, and that's what our body mounts an immune response to. You should know the MHC, the major histocompatibility complex, also known as the human leukocyte antigen. These are the cell surface markers which allow us to distinguish between self and non-self. Knowing the difference between MHC class 1 and MHC class 2, probably not all that important, but at this point, but knowing that I guess at least that MHC1 deals with your CD8 or your cytotoxic T cells, and MHC2 interact with CD4 or the helper T cells. Macrophages, to point out one of the important things that they do, they not only engulf and break things down, but it's the processing and displaying of antigens, which is really an important thing that you should know, the whole antigen presenting cell function. And that's really what will then activate the more specific immune response. T cells, the cell-mediated immunity, which includes your helper T cells and your cytotoxic T cells. These are the ones that are produced in the bone marrow. They mature in the thymus, and as opposed to your B cells, which are produced in the bone marrow, mature in the bone marrow, and they're part of the humoral immunity, which is the production of antibodies. The different immunoglobulins, the 5 immunoglobulins, I guess if I'm going to point out 3 of the 5 that are better to know than the others, IgG being the most common and being found in both the primary and secondary immune responses. IgM being the first to kind of rise in an immune response, and IgA being the one that oftentimes is in that first line of defense because it's found in the saliva and the tears and any of the mucous membranes. Probably remember something similar to this from last year, where we talked about the difference between natural and artificially acquired immunity, active versus passive. So again, I'll leave that, or you can, if you want to go through that in more detail, you have the option to do so. And knowing, you probably remember this from last year as well. It's the primary versus secondary immune response. So if you see antigen A, your body targets antigen A initially, the immune response is, whatever it is. But if it sees that antigen again in the future, then the immune response is much more rapid and much more intense. And the idea being that that's the idea of building up immunity, that you've seen it before, and it's also the principle of vaccination, is to kind of prepare you and put you in a position where you're all ready for that secondary interaction, and you can mount a very rapid and very massive response. Two things with regards to the immune system that I said were good to know, and it's the idea that it is not, although there are a number of components, they do not function independently. It is an integrated system, and the whole response is coordinated, and the more efficient the coordination between the different components, the more effective your immune response will be. And of course, the idea of regulating an immune response, because the immune response itself can cause damage to tissues, and regulating the whole response aims to not only rid the body of whatever the invading antigen might be, but also to reduce the damage to any surrounding healthy tissue as well. So, if it's not carefully monitored, then a lot more damage can result than what would have probably resulted from the initial infection. I wanted you to make a list of factors that could compromise the immune system, and I've made one for you. So I'm not going to go through these in detail, but you can take the time and sort of look through and see different things that could compromise how well and how effective our immune system is. And some of them, I think a lot of them do make sense or are fairly straightforward. So, you can take the time and look through those if you like. We talked about the two types of excessive immune responses. So the autoimmunity, which is where our body recognizes our own antigens, ourself, as foreign, and mounts a response that damages the tissues, versus hypersensitivities, where you have a normal immune response, but it's triggered by something that is not considered or not really dangerous. So it's a damaging response to a potentially harmless antigen. With regards to the different types of immune hypersensitivity reactions, there are 4 types. So we have the anaphylaxis or type 1 hypersensitivity, the cytotoxic hypersensitivity, immune complex, and the delayed or cell-mediated hypersensitivity. So there is a table in your textbook on page 199. Not a bad idea to at least look through the different types. I'm going to focus on type 1 anaphylaxis. It's one that I think you would probably encounter in the future, and this is where it's a systemic response. Enters the blood very directly, circulates rapidly. We're talking within a few, I think within about 15 minutes, it reaches its peak. Again, specific immune cells, basophils, mast cells, they release histamine, and then the histamine causes the damaging effects. So you get constriction of bronchioles, so you can't breathe. That you get sudden vasodilation, massive vasodilation, so you get a lot of fluid loss from the blood, and what could end up happening is you end up with a circulatory shock, where you have too much fluid flowing, blood flowing out of the, you have such widened blood vessels that the blood is unable to circulate to the body, and that's what actually causes death. And treatment, EpiPen, epinephrine. Why? Well, we talked about how it is both a potent vasoconstrictor and a bronchodilator, so it really counteracts the two major damaging effects of histamine. Autoimmunity, this is again where our body produces antibodies or autoantibodies, so antibodies against ourself, our own tissues. I talked about the four examples: systemic lupus, erythematosus, or lupus. This is one where you get chronic inflammation of multiple organs, so it includes things like the skin, the heart, the lungs, the joints, the brain. Very difficult to diagnose because of multiple organ involvement, but it does have a distinctive facial rash, which I'll show on the next slide. Not in all cases, but in some cases. MS is it's a demyelination of the nerve fibers, so you lose that coordinated nerve impulse function. Hashimoto's thyroiditis is an example of hypothyroidism, so you get decreased thyroxine production, which decreases metabolism. And rheumatoid arthritis is another example where the immune system targets the synovial membrane in the joints and causes inflammation, pain, and deformity in some cases of the joints. Factors to consider, we talked about genetic predisposition, so females are more likely to have autoimmune disorders than males, probably linked to MHC proteins, MHC proteins also play a role in this. So if you have certain MHC genes, then those are linked to a higher incidence of autoimmunity. Environmental factors, so being exposed to certain infectious agents, so certain bacteria, certain viruses might stimulate an autoimmune response in your body, and of course any other environmental stress, whether they're occupational or something you might be exposed to in your home, those could stimulate an autoimmune response as well. And of course, with regards to pharmacotherapy, to control autoimmunity, the main thing that you'll have to do is be on immunosuppressive drugs, and those in themselves can have side effects and complications. So corticosteroids are really used to try and control that autoimmune response. This is the distinctive facial rash, the butterfly type rash that a lot of people do have when they have lupus, but again, not everyone does. So immunodeficiency is when our body is not able to carry out its normal immune function. It could be something that you're kind of born with or develops from potentially a mutation, or could be something that was a developmental problem, and what ends up happening is that you are missing one or more components of the immune system, and you can't fight off infection nearly as well as somebody else. Versus secondary deficiency, which is what we call acquired immunodeficiency. So yeah, you could have poor nutrition. You might be on immunosuppressive drugs or chemotherapy. You could be infected with HIV, which could lead to immunodeficiency. So those are all examples of things that are not inherent. They're not something you're born with or genetically related. They just happen as a result of something that you encounter in your life. Focus a little bit on HIV. The reason that HIV is so damaging, how HIV actually causes AIDS, is that it targets the helper T cells, and if you know helper T cells, well, they coordinate the entire immune response. So without helper T cells, the immune system does not function. Now, what normally happens is over time, HIV slowly destroys your helper T cells, and from one person to another it varies as to what this time frame can be. But at some point the body loses so many CD4 cells or helper T cells that it becomes unrecoverable, and that's when you transfer from HIV infection to AIDS. And at that point, you're on a one-way path to to death, because it's not possible to sustain the body, and you don't end up dying of HIV. You end up dying of a common cold or a pneumonia in a lot of cases. For treating HIV, it's really targeting the virus's replication process, because it actually uses your own body cells, so it uses your helper T cells to replicate and make more HIV particles. So the drugs for treating HIV include ways to try and stop the virus from replicating inside your cells. And because it is a retrovirus, it has a very high mutation rate. You have to often be on multiple antiviral drugs, and I know the drugs are not cheap, and everybody responds somewhat differently. Some people, can live for an extended period of time and have very low levels of HIV, whereas others, even with the multiple drugs, the disease progresses much more rapidly. When we start talking about pathogens now, specific pathogens, a pathogen is really any organism which can cause a disease. And we are surrounded by pathogens, but our immune system does a pretty good job of protecting us. And if you want to take some time, just think of how in our everyday lives you might protect yourself against pathogenic organisms, what healthy living strategies you have. So you might want to include things like always washing your hands, probably very important for you, seeing as what your professional work environment is going to be like. And maintaining good health, so adequate nutrition. I mean, you could go through and come up with a few answers for those. I do want to take a bit of time just go through the whole principle of infection and how infection actually happens. So you usually start with a reservoir of some kind, so the where the infectious agent is present, how it leaves that environment or that location, so we call the portal of exit, how it's transmitted, which I'll talk about in a second, how it actually gains entry to a potential host or a susceptible victim, and then it carries out its function. So the whole reservoir, carrier, new host. This is not completely centered the way I wanted it to, but goes from its original site. It's carried to a certain either through a vector, or it can be carried through another individual, and then it ends up in a new host. Different modes of transmission. We can have direct contact. So this is where there's one individual has direct contact with another person. So it could be like having an infectious lesion, sexual intercourse. I mean, anything that involves direct contact from one carrier to a new host. Indirect contact is usually involves some sort of intermediary. So just as an example, if you have contaminated water, if you have like contaminated shellfish that somebody then consumes, that would be an indirect contact mode of transmission. Droplet is oral or respiratory. When you have respiratory or salivary secretions that contain the pathogens, get expelled from the body, and then they end up being inhaled directly by another person. Versus an aerosol, which is somewhat related to droplet, but this is where you have small particles that from the respiratory tract that remain suspended in the air, and they travel on air currents, and then they end up infecting a new host who inhale them. And then vector-borne transmission is where you might have an insect or an animal that is an intermediary host, like mosquitoes in West Nile virus or in malaria. wash your hands always. That's probably the best way to control transmission of infection. So microorganisms, we're going to talk about some different kinds. So bacteria, fungi, protozoa. Viruses are classified as microorganisms, but they're not really a microorganism because they're not really a living entity. Um, we can start with bacteria, and I'll move through these. Actually, I'll come back to bacteria in a minute. I want to get into something known as resident flora, and this actually is all about bacteria, but the actual bacterial structure and you know the difference between one bacteria and another, and how they cause infection. We'll talk about later, but there's something known as resident flora. So there are areas in our body where we actually have bacteria that are very helpful and they don't cause infection. So we have areas like the skin, the digestive tract, um, um, trying to think of other places. Those are two that kind of come to mind. So what the resident flora do is they might help break down food, like in our digestive tract. On our skin, they actually play a role in in preventing other bacteria, other potentially harmful bacteria, from establishing a population. And if you look at areas in the body where you shouldn't have any bacteria, shouldn't have any resident flora, those would include areas like the blood, the brain, the liver, um, areas where you wouldn't want any type of bacterial colony establishing itself. If we go through the infectious process, the whole host microbe environment interaction, um, we'll get into so the host itself, which could be us, the host characteristics. So having proper barriers, whether we are whether an individual host has any risk factors. So what's their nutrition like? How old are they? Are they on immunosuppressive drugs? Do they have any chronic illness? Um, um, are they or have they been immunized? So the role of immunization is important. That might make one host more susceptible than another. The environment plays a role, so depending on the climate and living conditions, the socioeconomic status, sanitation, I mean, all those things can affect how the infectious process proceeds. So in a much more or a much less sanitary environment, you're more likely to have infection than you are in a more sanitary location. If we looked at the microorganisms themselves, I'll just quickly go through what each one of these terms really relates to. So virulence determines how well a microorganism can infect the host. Adherence is the ability of the microorganism to stick to the host cell. So bacteria have projections known as pili that help them do this. Invasion is how they enter the cell. So vacuoles, for example. Toxins. Toxins are harmful to the cell, so exotoxins are something that's released by a living cell, so like a living bacterial cell, versus endotoxins, which are usually found in the cell membranes and are released when the cell dies. Bacterial enzymes, sometimes they have tissue-degrading enzymes. Some have anti-phagocytic factors, so they have an outside coating on some bacteria that prevent them from being phagocytosed, which makes it difficult for your immune system to deal with them. Endospores are highly resistant resting states of bacteria that really can sit in unfavorable conditions, and they can wait for favorable conditions to come along. They could sit there for a couple of weeks. Mobility, so being able to move, flagella, for example, on bacteria that allows them to move around, and having antimicrobial resistance. So some of them can produce enzymes that inactivate drugs, some of them create modified targets that are no longer susceptible to the drug, some have reduced permeability that prevents the drug from entering the bacteria, and some bacteria can actually actively pump out the drug. Oh, so that was a lot for that, so you may have to back up and listen to that again, but we'll move on now to kind couple things about host resistance. So, talked about interferons, the cytokines that are produced by the host to try and deal with a virus, for example, viral infection. So, and they also stimulate the immune system, and they're being more widely used now in treatments and cancer treatments, for example. Factors that decrease host resistance. So again, we've talked about what might compromise the immune system. Well, this is another. A lot of these are similar, because what makes a host more likely to be infected is probably similar to what compromises their immune system. So again, you can go through those on your own time. And we've we've already talked about the virulence and pathogenicity of microorganisms, but the whole threat, the superbugs. If you want to think maybe about what might be causing superbugs, and sometimes it's the excessive sanitary practices that we have that maybe allow only the strongest bacteria to survive. That might contribute to superbugs. I think one of the main reasons is people not taking all of their medication to completion. Again, making it a much more likely environment for the strongest bacteria to survive. Some universal precautions to try and control the transmission of infection. I think a lot of these make sense. Using disinfectants, antiseptics, proper precautionary practices with regards to travel, and sterilizing equipment, making sure immunizations are up to date. So again, I think most of these probably make sense. This kind of goes back to our Unit 1 with regards to terminology, the incubation period, the prodromal period, acute period, the convalescence, and I've thrown in septicemia/bacteremia as an example of an acute systemic infection. So again, important that you know the terminology, and you can kind of go through those on your own time. So here we are back to prokaryotes, which are the simplest living things. I guess the important things to know with regards to prokaryotes is really we're going to focus on bacteria. So I won't really spend much time talking about the different types of prokaryotes. Sorry, I went through that relatively quickly. I guess one thing you should know is that some of them can live in oxygen-rich and/or oxygen-poor environments, so they can survive in both. And some of them can switch from one type of survival mode to another depending on their environment. So that might be one good thing to know. Some disease-causing bacteria that I'll put up here that you can, again, you can take a bit of time and look at. So a number of these you've probably heard of, and I don't know if I'd ask you specifically to name the bacteria that causes a certain associated illness, but probably good that you at least are familiar with some of these. The structure of a bacterial cell or prokaryotic cell. I mean, it's a very simple structure. The core is made up of some sort of DNA material. It has a cell wall, and I mean, some of the structures, the pili, which allow the bacteria to stick to a host and carry out its function. The flagella, which allows it to move around. I mean, those are two important structures. And all of them have a glycocalyx in their cell wall, which is really we have it on all our cells as well, and it allows us to distinguish between what is self and what is not self. And depending on the type of bacteria, they're going to have a slightly different glycocalyx. But specifically with the cell wall, what you may want to know the difference between is gram-positive and gram-negative, and it really just has to do with slightly different protein structure, slightly different staining. But with regards to the different gram-negative organisms, I can give you a few. So strep, pneumonia, E. coli are two examples of gram-negative organisms. and gram-positive Staphylococcus aureus, which is a normal skin bacteria. Bacillus anthracis, or the anthrax bacteria, is another example of gram-positive. Generally speaking, gram-negative bacteria tends to be more dangerous than gram-positive bacteria, and I think it has to do with how our body reacts to the endotoxins that gram-negative bacteria have. We tend to have a more difficult time with gram-negative bacteria. There's some key points about prokaryotes, and again, you can look through those on your own time. I've asked this question many times: can a virus be killed? And the answer is no, it can't really be killed. It can be inactivated. The reason that it can't be killed is because it's not a living organism. It needs a host to survive. They can't be treated with antibiotics because they don't have a cell. The only way you can destroy them is really by destroying the cell that they live in, or the cell that they are replicating in, which tends to be the host cell. Some of them can stay latent or dormant for long periods of time. So herpes virus, for example, they could flare up and then go away and come back later on. We do call them parasites because they need a host cell to carry out their replication. And as they replicate and produce more viruses, they bust out of the cell, and that's when they actually cause damage to the cell. Viral structure, good to know the viral structure. So it's either a DNA or RNA core, and surrounded by a protein coat. So a very simple structure. They attach, they enter, they make copies, they bust out of the cell, they go look for another cell. That's really what viruses do, and the damaging effects really depends on the site of their target site. So if they adhere to something like skin cells, maybe not as damaging as if they attach to something like your T helper cells. Some examples of virus: so hepatitis, herpes, Ebola, that's the one that was in that first diagram, the flu, German measles. Retrovirus, I briefly talked about retroviruses. It's actually wrong. I should say contains an RNA core and an enzyme to convert the RNA into DNA, and it's this process that makes retroviruses so highly mutating, which is why they're difficult to protect against, and HIV is a good example of a retrovirus. As far as viral replication goes, again, you've probably seen diagrams of this before, but we attach to a host and it enters, so penetration, and then the virus uncoats, uses the host to make new viral parts, assembles the new virus parts into new virus particles, and then they release many new viruses, and usually ruptures the host in the process, and they go look for a new cell to invade and destroy. Some other microorganisms that you should be familiar with are fungi. Fungi live everywhere, usually on dead organic material. They can cause something known as mycoses or mycotic infections. So tinea pedis, athlete's foot, thrush, histoplasmosis in the lungs. Those are all examples of fungal infections. Protozoa are complex unicellular organisms that are they're motile. They have no cell wall. They have irregular shapes and can change at times. Some examples of common protozoa are malaria is an example of a plasmodium, a type of protozoa. Amoeba, a certain amoeba, amoebic dysentery causes severe diarrhea, and trichomoniasis as well. So those are 3 examples for you. So even having more microorganisms that you should kind of be familiar with. Protozoa I just talked about. The other, there are 4 main families, but if we talk about the worms, first of all, a lot of these parasites cause infections and enter humans via contaminated water, through the skin, or infected insects. So, and these are found all over the world. Some of them are more common in certain places based on sanitation and nutrition levels, clean water, things like that. The worms, so the parasites that are considered worms, they're usually diagnosed by finding the eggs in stool specimens, and if you have large numbers, they can cause severe anemia. And I've already kind of mentioned malaria as an example. So malaria uses mosquitoes. The Plasmodium uses mosquitoes as a host to then transfer from one individual to another. And again, a parasite, because it needs a host to replicate and survive, it cannot survive on its own. I don't really talk much about prions, but you probably remember, or you may remember, the mad cow disease outbreaks. Those are examples of prions. They're proteinaceous infectious particles, which are they're not viruses, not bacteria, they're not fungi. They just happen to be like a protein agent which accumulates in the brain, causes brain wasting. Creutzfeldt-Jakob is kind of the human version of mad cow disease. And even more microorganisms to kind of finish off on. Chlamydia, they're a type of primitive bacteria. One's infectious, one isn't. The sexually transmitted version is the infectious version. Rickettsia are tiny gram-negative bacteria that are transmitted by certain vectors such as ticks or lice. Typhus fever is an example of this type of microorganism. Again, one that thrived in non-sanitary conditions. And mycoplasmas are a type of bacteria that really have no cell walls. They're kind of like bacteria, but they're missing that major structure, and they are common causes of pneumonia. So all three of these have some similarities to bacteria and viruses, those being that they replicate by binary fission, and they need living cells for reproduction. So they can't live by themselves, but they tend to follow a replication pattern that is similar to bacteria. So I tried not to go off on tangents and talk too much about irrelevant too many irrelevant things in this presentation, just to give you, to limit the amount of time that you have to go through this stuff. So I hope this wasn't too dry for you, but again, you can go back through it on your own time, and just remember the test, all the components of the test, and the breakdown. And if you go through this presentation, if you go through the other notes that you have, and you do the review questions, I think you're going to do just fine on the test, and we will see you on Monday.